Good. Well, good afternoon, everybody. As Marissa said, I'm Michael Benatar. I'm a neurologist at the University of Miami, and I've had a long-standing interest in studying pre-symptomatic ALS and trying to get to the point of early intervention in ALS. And I'm excited to have the opportunity today to talk to you a little bit about the ATLAS study and to give you some information about this. So the ATLAS study is a clinical research study that's looking to enroll people who carry certain variants in the SOD1 gene, but who are still pre-symptomatic, who've not yet developed ALS. And the goal of the study is for me to provide you with some more information about the study in case you or somebody whom you know might have an interest to learn more or to participate in this study. So here's what I'm going to try to cover. I'm going to start out with just a little bit of background about clinical research and what a clinical research study is and some different types of clinical research studies. I'll talk very briefly about genetics and biomarkers in ALS, really as those pertain to the design and implementation of the ATLAS study. I'll talk about the investigational drug that's being used in this study. I'll then get into the meat of the presentation, which is really the details of the ATLAS study. And then end up with pointing you to some resources that may be helpful as you're trying to acquire more information or consider whether this is a study for you or somebody that you know. So why don't we start out with what is a clinical research study? And I'm guessing for most of the audience on this webinar, this is something that you already know. Broadly speaking, we think about clinical research as being either observational or interventional. And interventional studies are often called a clinical trial. And I'm going to focus on the idea of a clinical trial here today because that's fundamentally what ATLAS is. So a clinical trial or an interventional clinical research study is really a scientific investigation that aims to answer important questions about the drug that's being investigated. These are questions such as, is it safe? How well does it work? Does it work? Which dose is best? What are the side effects of the drug? As I'm sure you understand, for any study, and certainly for clinical trials, every study requires review and approval by an ethics committee or an institutional review board. And this is standard practice for clinical trials and clinical research to ensure that the rights and well-being of participants are protected, that there are no unnecessary risks involved, and that the study is scientifically rigorous and will answer important unanswered questions. And really all investigational drugs have to be tested in this way through a clinical trial before they can be assessed or potentially approved by a regulatory authority. And that needs to happen before an investigational drug can then become approved and prescribed by a physician. So by way of background then to the ATLAS study, I wanted to highlight, there are many challenges to ALS therapy development or ALS drug development, but there are two that I want to draw your attention to, which have really been fundamental to our thinking about the ATLAS study. So the first is if we think about ALS more broadly, in general, we have a limited understanding of the cause or causes of ALS. And again, I think this audience knows that there are many potential causes of ALS. We know there are many different genes and there are many causes that we've not yet identified, but often not knowing the cause of disease makes it hard when we're trying to develop effective therapies. But the other problem, and this is sort of almost uniformly the case in clinical trials in ALS, is that we face this challenge, this problem that there's often a long delay in diagnosis and then from diagnosis to treatment. And we worry that bringing treatments to bear late in the course of disease may be much less effective and potentially so than initiating treatments much earlier. So let's then talk a little bit about some information, both genetics and biomarkers, that are relevant to some of these considerations. So I've alluded to the idea that there are many genetic causes of ALS, and the SOD1 gene variants or mutations in the SOD1 gene are one of those important causes. SOD was the first genetic cause of ALS that was identified, and currently our best estimates that about 2% of people living with ALS have a disease-causing variant in the SOD1 or superoxide dismutase gene. And it's important to underscore that we see SOD1 mutations that can be disease-causing, both in people who have a family history of ALS, so in what we call familial ALS, but also in people who don't have a family history of ALS, what's sometimes called sporadic ALS. So there isn't a neat alignment between a genetic cause and familial or sporadic. Important to understand that. But irrespective of whether there's a family history or not, what we think happens is there's this disease-causing variant in the SOD1 gene, and that leads to a misfolded SOD1 protein. So the gene is the DNA that is the genetic blueprint, and that causes the body to make proteins. And when there's a mutation in the SOD1 gene, that leads to a misfolded SOD1 protein. And through mechanisms that are not well understood, we think that that leads to degeneration of nerve cells. And that's the progressive degeneration and progressive weakness that we see in ALS. So in SOD1 ALS, for example, we understand the cause of ALS, even though that's only in 2% of the population. So I need to introduce you, if you're not already familiar, with two concepts. The one is penetrance, and the other is what's sort of referred to as disease progression. And both penetrance and disease progression, how quickly the disease unfolds, vary enormously, even amongst people who have a mutation in the SOD1 gene. There are, in fact, over 200 different variants that have been described in the SOD1 gene, some more common than others. But let's talk about what penetrance means. So penetrance is getting at the idea that not everybody with an individual SOD1 variant will develop ALS in their lifetime. So penetrance refers to or describes the proportion of people with that SOD1 variant or with a genetic variant who will develop disease. If penetrance is 100%, everybody with the gene will develop disease. If penetrance is 50%, 50% of people with the gene will develop disease. So not all SOD1 variants have the same penetrance. It's also true that not all SOD1 variants produce the same clinical course of disease. So how quickly disease unfolds and progresses varies tremendously. Some SOD1 variants are known to be associated with much faster progressing disease, and some with much slower progressing disease. And for some, it's variable. The same mutation or variant may sometimes lead to faster disease and some to slower disease. But these are both important because both penetrance and the anticipated rate of disease progression factor into eligibility for the ATLAS study. So let's then shift from talking about some genetics to talk a little bit about biomarkers. So I think, again, this audience knows that biomarkers are things that we can measure that tell us something about the underlying disease. A biomarker can be used in different ways, but it might tell us something about how somebody is at risk for disease, what the future course of disease is, or how an individual or a disease might respond to a particular treatment. One example of a biomarker is this protein called neurofilament light, or NFL. And this is a protein that's one of the building blocks of nerve cells. Here you see the nerve cell and its axon. And when the nerve cell is damaged and degenerates, those building blocks of the neuron, that neurofilament, are released into the spinal fluid, make their way into the blood, and we can measure neurofilament in spinal fluid or blood. And we think that neurofilament has the potential to be a biomarker. And this has been investigated or studied by many groups. And one of the things that we have found is that neurofilament light levels seem to increase in the 6 to 12 months before the onset of signs or symptoms of ALS. And this is in a subset of people who carry an SOD1 variant that is associated with rapid disease progression and, in fact, high penetrance. So I want to just sort of emphasize that again, that NFL as a biomarker can be used in different ways, but the important observation is that amongst some people who carry certain SOD1 variants, we may see an increase in neurofilament in the 6 to 12 months before clinical manifestations of disease emerge. So with that background then, let me tell you a little bit about the ATLAS study, which aims to evaluate the impact of starting an investigational drug prior to the onset of ALS symptoms or signs, prior to the emergence of what we think of as clinical ALS. And the investigational drug will be given based on a biomarker, in this case neurofilament light, as a marker or as evidence that the disease, that there is some underlying disease activity. So let me sort of dive into that a little bit more. But before I do, I want to tell you about the investigational drug that's being used here. and this is a drug that's known as tofersen. And this drug is also called BIIB067, but tofersen is the name that I think is now most commonly used. So tofersen is an investigational drug. It's not approved by any regulatory authority at the moment, and its safety and efficacy have not been established. It's important to understand that this is a drug that's under investigation at the moment. So what is tofersen? It's a drug that's designed to target SOD1. This is important, right? I said that often we don't know the cause of ALS, but when you have an SOD1 mutation, we know the cause, and that leads to this abnormal misfolded protein. And tofersen is designed to target that SOD1. And so what it does through a mechanism that I don't need to get into the details of, it leads to a reduction, or it's expected to lead to a reduction in SOD1 protein levels. And there's good reason to believe this based on how it works, based on some preliminary data. And there's also the hope that this would potentially slow disease progression. So how is tofersen administered? It's administered as an intrathecal injection. So what that means is a trained clinician, typically a neurologist, will perform a lumbar puncture, so put a needle into the base of the back after some local anesthetic. They'll draw off some spinal fluid, and then they inject the drug, the tofersen, into that intrathecal space, into the spinal fluid space. And the way this is given is there are typically 3 loading doses every 2 weeks. And after that, the maintenance therapy with this is that the drug is given intrathecally every 4 weeks. And the most common side effects are those associated with a lumbar puncture, and these are often pain where the needle is inserted, and sometimes a headache after the lumbar puncture. And this is common even when we don't give an investigational drug. Headaches are not that uncommon after a spinal tap. So this is a busy slide. I'll spend a little bit of time going through this and try to sort of work you through it. So there have been studies looking at tofersen. It's been evaluated in people living with SOD1 ALS in a phase 3 study, as well as an open label extension. That phase 3 study was a double-blind placebo-controlled trial, and then an open label extension. And of course, just to draw the distinction, ATLAS is not for people living with ALS and an SOD1 mutation. It's people who have a mutation who have not yet developed ALS. But having said that, the background here is that tofersen has been evaluated in a phase 3 study. And before I talk to you about what was found, I want to just clarify a few sort of concepts that exist in the clinical trials world. So the first is this concept of a primary endpoint. So when we design a clinical trial as an experiment, we specify the primary endpoint, which is the planned outcome measure that's most important for evaluating the effect of that intervention or the treatment. But we also typically include in clinical trials what are called secondary endpoints. These are planned outcome measures that may not be as important as the primary outcome for evaluating the effect of the intervention, but these are still of interest and can teach us something about how the drug may or may not be working or isn't safe. And then there are often exploratory endpoints. These are clinically important events that are expected to occur perhaps too infrequently to show an effect, but they're included because we want to learn new things, develop new hypotheses. And although we can't draw definitive conclusions from these exploratory endpoints, they can generate new hypotheses or ideas of things that we then need to study in future studies. And I'll come back to these concepts both as I describe the results of this phase 3 study, but also as I describe the ATLAS study to you. So the phase 3 study was called VALOR. You may have heard that name, VALOR, sorry for my accent. And while that study did not meet the primary endpoint of a change from baseline to week 28 in the revised ALS functional rating scale, that's a rating scale that measures functional ability with activities of daily living in ALS, there were trends, there did appear to be trends favoring tofersen across multiple secondary and exploratory measures of both biological activity and clinical function. And if one looks at the combined evaluation of data from the phase 3 study, the double-blind, and its open-label extension, it suggests that there may be a benefit from earlier treatment than later treatment with tofersen, and that's relevant to ATLAS too. Now, the most common side effects in people with receiving tofersen were procedural pain, so at the site of the lumbar puncture, headache, pain in the extremities, fall, and I've mentioned back pain. And most side effects were mild to moderate in severity. There were some occasional serious neurological side effects. These were seen in 4.8% of participants who were on tofersen, including 2 patients who developed myelitis, which is an inflammation of the spinal cord. There was also one death reported in the tofersen group in the phase 3 study, but the investigators determined this not to be related to tofersen. So if you want to learn more about this, talk to your local neurologist or the investigator that you work with, and they can help walk you through this. These results have been presented. So let's then talk about the ATLAS study. So what are we hoping to learn through the ATLAS study? The primary objective, you'll remember I've spoken about the primary objective before, is to evaluate the efficacy of tofersen in participants carrying an SOD1 gene variant who do not yet have signs or symptoms of ALS, but do have a neurofilament level above a predefined threshold. And the way we'll get at this primary objective is we will measure the proportion of participants who develop signs or symptoms of ALS within 12 months of Part B baseline. And Part B is randomization to receive the drug. And that'll become clear in a moment what I mean by Part B, because there's a lead-in period where people are just being watched, but then in Part B, people are randomized to receive tofersen or placebo. And the proportion of people with signs or symptoms of ALS from the first 12 months, that will be the primary measure of efficacy. Secondarily, we want to evaluate the safety, tolerability, and other effects of tofersen when initiated before emergence of clinical ALS, as opposed to at the time that symptoms and signs of ALS do emerge. And you'll see from the schema that I'll show you in a moment how we'll be able to do that. And safety measures will include things like the incidence of side effects, serious side effects during the treatment period. So before I come to more details of the study, procedures and design and how it looks, let me tell you a little bit about eligibility, because this is not for everybody. So firstly, you need to be at least 18 years of age at the time of informed consent. Secondly, you need to be confirmed as a carrier of a certain SOD1 gene variant, and these are a subset of variants that are associated with high or complete penetrance and are known to be associated with rapid disease progression. Importantly, you must not have any clinical signs or symptoms that definitely indicate the onset of ALS at the time of screening. And at least initially at screening, to get into that observational period, you need to have a low plasma neurofilament level. It needs to be below the threshold at the screening period. Now, other eligibility criteria will also apply, but these are sort of the core ones that we start with. So I've referred to sort of different parts of the ATLAS study. Let me walk you through this because there really are 4 parts. So after study screening and enrollment, there's Part A, which is a natural history study. And during this period, people come to the study center at the beginning to be enrolled. And then they're followed monthly with blood draws in your home to measure neurofilament levels. There's no study treatment during this time. We're simply monitoring neurofilament levels. And we anticipate enrolling about 150 people in this phase. Now, if during this course of monitoring neurofilament, we see that plasma neurofilament levels rise above a predefined threshold that the study has set, and if a person is still pre-symptomatic, there's still no symptoms or signs of ALS, then they can be evaluated and, if eligible, randomized 1-to-1 to receive tofersen or placebo. And as I said before, this is given 3 loading doses 2 weeks apart and then monthly. Now, this has to be done at the study center, so you have to travel to the study center for these. And then that drug continues until either the disease doesn't develop or signs and symptoms of disease emerge. And if they do, and if the participant is interested, they have the opportunity to move over into Part C, which is open label tofersen. So under the scenario, the latest somebody receives active drug is at the point in time of developing signs or symptoms of ALS. Now, it's also possible, as we're following people, that someone might develop signs or symptoms of ALS without the neurofilament level having gone up. And if that occurs, people go into this Part D, which is open label tofersen. And Part D or Part B and Part C combined, these may last up to 2 years in duration. So it's a little complicated, but if you do want to know more, and I'll point you to some resources, take a look at it, and I think it's relatively self-explanatory. So study visits, I've spoken a little bit about these, and I've sort of mentioned that the baseline day 1 in Part A needs to be at the study center. But then the follow-up visits when we're monitoring neurofilament, this is done in your home. And these assessments during Part A are limited to blood and urine collections, home digital assessments, and you're reporting any signs or symptoms of ALS, if any were to emerge. And I think there are a total of 58 study visits that may occur in your home. If one, and yes, I said these occur every 28 days. If one then gets into part B or C or D, then these are occurring at the study center, and there are potentially 34 visits here that happen initially every 2 weeks and then every month. Now, a critical part of the study is obviously genetic testing and genetic counseling. We would never do genetic testing without appropriate counseling. And the way in which this is done is with pre-test counseling. So first you meet with a genetic counselor, you learn about the potential pros and cons of going through genetic testing, understanding social support systems, psychological impact of testing, implications potentially for health insurance, for life insurance, for employment discrimination. So it's important that there's pre-test genetic counseling before one decides whether to undergo genetic testing. You then undergo SOD1 genetic testing if you decide to proceed, and then post-test genetic counseling to receive the result, whether that result shows an SOD1 variant or it doesn't. And important to say that these counseling sessions will cover not just genetics, but also discussions about neurofilament light and the potential implications of a low level, of a high level, of a level that becomes elevated over time. And importantly, because we know that this can be stressful to receive this information, and not the genetic information, but neurofilament levels will change over time. And so additional counseling sessions are available upon request throughout the course of the study. And so if you're enrolled, you just have to let your study investigator or coordinator know, and there are additional counseling resources available. So just a few more words about genetic testing, because this is a very important thing to think about. There's no right or wrong answer here about whether one should undergo genetic testing if you're concerned about whether you carry an SOD1 variant. What's right is what's right for you in the context of your own individual life, your family, your friends, your context. And those are things that you should discuss with your physician, with the study center, with the genetic counselor. There are potential benefits if you learn this information and know whether you do or don't carry a mutation. You can be more perhaps proactively involved in planning your health and planning your future things in your life. But there are also potential risks. You know, if you carry a gene mutation that may be relevant to family members, siblings, parents, children, there's also at least the potential for genetic discrimination, so discrimination based on genetic results in the employment, insurance, or disability arena. And there are safeguards in place, and there's strict separation of medical and research records, but you need to understand these issues, consider them, understand the protections, and these are all things to discuss both with your physician and with the genetic counselor. Another thing to underscore, and this is true not just for ATLAS, but really for any clinical study and certainly for any clinical trial, is that participation is always voluntary. And even if you've decided to participate, you can withdraw at any stage. That option is always open to you. You're in control. So people are willing to volunteer for a clinical research study to make it possible for researchers, for scientists to evaluate investigational drugs and to try to develop therapies. And people take part in clinical studies for different reasons. Sometimes they hope to get a benefit themselves, although they may or may not. Sometimes it's for altruistic reasons to help people in the future with this disease. So everybody has their own set of reasons. And so choosing to take part again is always voluntary, and you can withdraw at any time and for any reason. So there are many sites that are participating in this around the world. Here are the active sites in the US. You can visit clinicaltrials.gov for more information, but I believe that all of these sites are currently active, and so you could attend at any of these to be considered for the study. Now, we know not everybody lives in the vicinity or in the same city as one of these sites, and so you may be eligible to receive support for travel and help with coordinating that travel and reimbursing it, not just for you, but for a companion as well. And it's important to say that neither participants nor the insurance provider will be charged for the costs of the tests or procedures performed in the trial. And you can find out more from your local center depending on where you are, whether flight or ground travel is appropriate or supported for you. So I want to finish off then by just pointing you to some resources. There is a website, alsatlasstudy.com, and you can take a survey and see if you might qualify. So I've mentioned the study website and the pre-screening questionnaire. There's clinicaltrials.gov, and you can search for this identifier or search for the ATLAS study. And you can also contact the US Biogen Clinical Trial Center or your local doctor who might have information about this study. And so I think I will stop there, and I'm sorry we're not taking questions, but really do hope that this has been helpful and encourage you to ask questions, go online, learn more, and if this is right for you, then to participate as you see fit. So thank you very much.